This video is about some sensory receptors. Major sensory receptors. Learning objectives include classifying the sensory receptors and give examples, enumerate the properties of receptors, describe the labeled line principle and the law of projection, along with some major terms. Our perception of signals within our bodies and of the world around us are mediated by a complex system of sensory receptors that detect such stimuli such as touch, sound, light, pain, cold, and warmth. The purpose of this chapter is to discuss the basic mechanisms by which these receptors change sensory stimuli into nerve signals that are then conveyed to and processed in the central nervous system. Types of sensory receptors and the stimuli they detect. Mechanoreceptors, which detect mechanical compression or stretching of the receptor or of tissue adjacent to the receptor. Then thermoreceptors, which detect changes in temperature, with some receptors detecting cold and other warmth. Nociceptors, pain receptors, which detect physical or chemical damage occurring in the tissue. Electromagnetic receptors, which detect light on the retina of the eye. Chemoreceptors, which detect taste in the mouth, smell in the nose, oxygen level in the arterial blood, osmolarity of the body fluids, carbon dioxide concentration, and other factors that make up the chemistry of life. In this chapter, we discuss the function of a few specific types of receptors, primarily peripheral mechanoreceptors, to illustrate some of the principles by which receptors operate. Other receptors are discussed later. So, differential sensitivity of receptors. How do two types of sensory receptors detect different types of sensory stimuli? The answer is by differential sensitivities. That is, each type of receptor is highly sensitive to one type of stimulus for which it is designed, and it is almost non-responsive to other types of sensory stimuli. Thus, the rods and cones of the eyes are highly responsive to light, but are almost completely non-responsive to normal ranges of heat, cold, pressure on the eyeball, or chemical changes in the blood. The osmoreceptors in the supraoptic nuclei in the hypothalamus detect minute changes in the osmolarity of the body fluids, but have never been known to respond to sound. The osmoreceptors of the supraoptic nuclei in the hypothalamus detect minute changes in osmolarity of the body fluids, but never known to respond to sound. Pain receptors in the skin are almost never stimulated by usual touch or pressure stimuli, but do become highly active the moment tactile stimuli becomes severe enough to damage the tissue. Modality of sensation, the labeled line principle. Each of the principal types of sensation that we experience, pain, touch, sight, sound, and so forth, is called a modality of sensation. Yet, despite the fact that we experience these different modalities of sensation, nerve fibers transmit only impulses. Therefore, how do the nerve fibers transmit different modalities of sensation? The answer is that each nerve tract terminates at a specific point in the central nervous system, and the type of sensation felt when a nerve fiber is stimulated is determined by the point in the nervous system to which the fiber leads. For instance, if a pain fiber is stimulated, the person receives pain regardless of what type of stimulus excites the fiber. The stimulus can be electricity, overheating of the fiber, crushing of the fiber, or stimulation of the pain nerve endings by damage to the tissue cells. In all these instances, the person perceives pain. Likewise, if the touch fiber is stimulated by electrical excitation of the touch receptor, or in any other way, the person receives touch because touch fibers lead to specific touch areas in the brain. Simultaneously, fibers from the retina of the eye determines the vision areas of the brain. Fibers from the ear terminate in the auditory areas of the brain, and temperature fibers terminate in the temperature areas. This specificity of nerve fibers for transmitting only one modality of sensation is called the labeled line principle. And the impression of the specific stimulus arising when the specific nerve fiber is stimulated anywhere along its path to the brain is called the law of projection. Once again, The impression of the specific stimulus arising when the specific nerve fiber is stimulated anywhere along its path to the brain is called the law of projection. Law of projection and labeled line principle. Next topic, transduction of sensory stimuli into nerve impulses. Local electrical currents at nerve index receptor potentials. All sensory receptors have one feature in common. Whatever the type of stimulus that excites the receptor, it immediately affect it to the change in membrane electrical potential of the receptor. This changing potential is called a receptor potential. It's a change in the membrane electrical potential. So there will be a receptor potential change. Mechanism of receptor potentials. Different receptors can be excited in one of several ways to cause receptor potentials by mechanisms of receptor potentials. Different receptors can be excited in one or several ways to cause receptor potentials by mechanical deformation of the receptor, which stretches the receptor membrane and opens ion channels, by application of a chemical to the membrane, which also opens ion channels, by change of the temperature of the membrane, which alters the permeability of the membrane, and by the effect of electromagnetic radiation, such as light or retinal visual receptor, which either directly or indirectly changes the receptor membrane characteristics. Once again, different receptor can be excited in one of the many ways to cause receptor potentials. Mechanical deformation, then chemical to the membrane, change of the temperature of the membrane, permeability change of the membrane, electromagnetic radiation. These are the various methods by which mechanism of receptor potential change. So there's four means of exciting receptors. The third one I mentioned, that permeability of the membrane is to a greater extent related to the temperature change. So that is considered as one point. Which are the 3 points? Mechanical deformation, chemical to the membrane, change in the temperature, and electromagnetic radiation. These four means of exciting receptors correspond in general to the different types of known sensory receptors. In all the instances, the basic cause of change in membrane potential is change in membrane permeability of the receptor, which allows ions to diffuse more or less readily through the membrane and thereby to change the transmembrane potential. Maximum receptor potential amplitude. The maximum amplitude in most sensory receptor potential is about 100 MV, but this level occurs only at an extremely high intensity of sensory stimulus. This is about the same maximum voltage recorded in action potentials and is also the change in voltage when the membrane becomes maximally permeable to sodium ions. Relation of the receptor potential to action potentials. When a receptor potential rises above the threshold for eliciting action potentials in the nerve fibers attached to the receptor, action potentials occur. More receptor potential rises above the threshold level, the greater becomes the action potential frequency. Receptor potential of the Pacinian corpuscles, an example of a receptor function. Pacinian corpuscle has a central nerve fiber extending through its core. Surrounding this central nerve fiber are multiple concentric capsule layers, and thus compression anywhere in the outside of the corpuscle will elongate, indeed, or otherwise deform the central fiber. Central fiber of Pacinian corpuscles, after all capsule layer but one have been removed. The tip of the central fiber inside of the capsule is unmyelinated, but the fiber thus become myelinated, the blue sheet known in the figure. So shortly before leaving the capsule to enter the peripheral sensory core. Receptor potential is produced in the Pacinian corpuscle, small area of the terminal fiber that has been deformed by compression of the corpuscle and ion channels have opened the membrane, allowing positively charged sodium ions to diffuse into the interior of the membrane. This action creates increased positivity inside the fiber, which is the receptor potential. The receptor potential in turn induces a local circuit of current flow known as and local circuit of current flow through the spreads to the nerve fiber. At the first node of Ranvier, which lies inside the capsule of the Pacinian corpuscle, the local current flow depolarizes the fiber membrane at this node, which then sets off typical action potentials that are terminated along the nerve fiber toward the central nervous system. And relation between stimulus intensity and the receptor potential. Changing amplitude of the receptor potential caused by progressively stronger mechanical compression, increased stimulus strength applied experimentally to the central core of Pacinian corpuscle. The amplitude increased rapidly at first, but then progressively less rapidly at high stimulus strength. In turn, the frequency of repetitive action potentials transmitted from sensory receptors increase approximately in approximation of the increase in the receptor potential. Putting this principle together with the data, one can see that very intense stimulation of the receptor causes progressively less and less additional increase in number of action potentials. This exceedingly important principle is applicable in almost all sensory receptors. It allows the receptor to be sensitive to very weak sensory experience and it not reach a maximum firing rate until the sensory experience is extreme. This feature allows the receptor to have an extreme range of response from very weak to very intense. Adaptation of receptors. Another characteristic of all sensory receptors is that they adapt either partially or completely to any constant stimulus after a period of time. That is, when a continuous sensory stimulus is applied, the receptor responds at a high impulse rate at first and then at a progressively slower rate until finally the rate of action potential decreases to very few or often not at all. Pacinian corpuscle adapts very rapidly, but here receptors adapt within a second or so, and some joint capsules and muscle spindle receptors adapt slowly. Some sensory receptors adapt to far greater extent than do others. For example, the Pacinian corpuscle adapted to extinction within a few hundredths of a second, and the receptors at the bases of the hairs adapted to extinction within a second or more. It is probable that all other mechanoreceptors eventually adapt almost completely, but some require hours or days to do so. For each reason, which reason they are calling non-adapting receptors. They are called non-adapting receptors. The longest measured time for almost complete adaptation of the mechanoreceptor is about 2 days, which is the adaptation time for many carotid and carotid and aortic baroreceptors. However, some physiologists believe that these specialized baroreceptors never fully adapt. Some of the known mechanoreceptors, the chemoreceptors and pain receptors, for instance, probably never adapt completely. Nerve fibers that transmit different types of signals and their physiological classification. Some signals need to be transmitted to and or from the central nervous system extremely rapidly, otherwise the information would be useless. An example is the sensory signals that apprise the brain of the momentary positions of the legs at each fraction of a second during running. At the other extreme, some types of sensory information such as the depicting prolonged anchoring pain do not need to be transmitted rapidly, and the slowly conducting fibers will be enough. The greater the diameter, the greater is the conducting velocity. The larger the diameter, the greater the conducting velocity. The range of conducting velocity is between 0.5 and 120 m per second. Then general classification of nerve fibers. General classification and a sensory nerve classification of the different types of nerve fibers. In the general classification, the fibers are divided into two types, A and B, and type A fibers are further divided into alpha, beta, and gamma and delta fibers. Type A fibers are typical large and medium-sized myelinated fibers of spinal nerve. Type C fibers are the small non-myelinated nerve fibers that conduct impulses at low velocities. And C fibers constitute more than one half of the sensory fibers in most peripheral nerves, as well as the postganglionic automatic fibers. The sizes, velocities of conduction and function of the different nerve fiber types are also very important. Note that a few large myelinated fibers can transmit impulses at velocities as great as 120 m per second, covering a distance that is longer than a football field in 1 second. The smallest fibers transmit impulses as slowly as 0.5 meter per second, requiring about 2 seconds to go from the big toe to the spinal cord. An alternative classification used by sensory physiologists. Certain recording techniques have made it possible to separate the type A alpha fibers to two subgroups, like group LA fibers from the ingulospinal endings of muscle spindles, then group 1B fibers from Golgi tendon organs, then group 2 fibers from most discrete cutaneous tactile receptors and from the flower spray endings of some muscle spindles. Group 3 fiber carrying temperature, crude touch, and pricking pain sensations. Group 4 unmyelinated fibers carrying pain, itch, temperature, and crude touch sensations. And transmission of signals of different intensity in nerve tracts, spatial and temporal summation. One of the characteristics of each of the signal that always must be conveyed in signal intensity, for instance, the intensity of pain. The different gradations of intensity can be transmitted either by using increased number of parallel fibers or by sending more action potentials along a single fiber. These two mechanisms are called respectively spatial summation and temporal summation. Spatial summation. Phenomenon of spatial summation, whereby increasing signal strength is transmitted by using progressively greater number of fibers. When a small portion of skin is stimulated, section of a skin innervated by a large number of parallel pain fibers. Each of these fibers arborites into hundreds of minute free nerve endings and serve as pain receptors. The entire cluster of fibers from one pain fiber frequently covers an area in the skin. This area is called a receptor field of that fiber. The number of endings is large in center of the field but diminishes towards the periphery. One can also see from the figure that arborizing fibrils overlap those from other pain fibers. Therefore, a pinprick of a skin usually stimulates endings from many different pain fibers simultaneously. When the pinprick is the center of the receptor field of the particular pain fiber, the degree of sensation of that fiber is much greater, far greater than when it is the periphery of the field, because the number of free nerve endings in the middle of the field is much greater than at the periphery. Thus, the lower part shows three views of the cross section of the nerve bundle leading from the skin area. To the left is the free stimulus and only a small single nerve fiber in the middle of the bundle stimulated strongly, represented by a red colored fiber. Then, whereas several adjacent fibers are stimulated weakly, the other two views of the nerve cross section between the effect of moderate stimulus and strong stimulus with progressively more fibers being stimulated. Stronger signals spread to more and more fibers. This process is known as spatial summation. Then temporal summation, a second means for transmitting signals of increasing strength is by increasing the frequency of nerve impulses in each fiber. This is called temporal summation. Transmitting signals from increasing strength by the increasing the frequency of nerve impulses from each fiber. Summation of the properties of receptors. Differential sensitivities, ordered projection to the cortex labeled line, graded responses, and adaptable changes.